Ophthalmology and the Director of Research, and the E. Ronald Salviti, right, Endowed Chair and a Professor of Ophthalmology at the University of Pittsburgh. John got his PhD in biochemistry from Ohio State. He then went to postdoc at Baylor. In 1999, he went to the University of Oklahoma, where he was tenured. And in 2011, he decided to move to the University of Florida, where he was the Francis M. Bullard Eminent Scholar and Chair in Ophthalmic Sciences in the Department of Ophthalmology. In 2022, he decided to come back east, God knows why, and up north to the University of Pittsburgh. For those of you who don't know John, and I think I've known John probably most of my career, he's an expert in retinal degeneration. His research focuses on understanding the molecular mechanisms behind vision loss, whether it be genetics or inherited retinal diseases, age-related macular diseases. He's worked a lot on neuroprotectins, cytokines, and metabolism, and I'm sure all of us know about his work on AMPK and metformin. So I think your title is very apropos to what is currently swirling at NIH. So without further ado, John, and stress-induced resilience.
We all need a little of that right now. I don't know about your emails, but my inbox blew up on Friday and it hasn't really stopped, but we'll get there. Thank you for inviting me. I really am excited to be here. There's so many friends here to get to see and visit with again. And, you know, it really is an amazing place. And, you know, we're still in the same state, although it takes hours to get here. But it is a wonderful opportunity to visit with you and to meet and discuss science with you. You know, I just moved to Pittsburgh and I moved December of 2022, and I had to explain to people I wasn't crazy to move from Florida to Pittsburgh in December. But actually, I did it for very good reasons. It is really a very, very fast-growing institution. Dr. Sahal is an amazing chair there, and it really is a great research environment that I'm just thrilled to be working there. So you get to come visit in Pittsburgh. We'll show you around. It's a beautiful city if you've not been there, and we have a beautiful new Vision Institute to show off. That it's really a great place to work. It's some great colleagues there. So the science is really amazing. So I'm just thrilled to be there. And I need to advance. So first I get going, I would say, you know, teamwork is dream work. Really believe that. I don't do a lot of work myself. I've actually been fortunate to have really some fantastic students working with me in the past. Almost all of the work I'm going to talk about today, it's their work and they're responsible. And I may not always give them credit, so I want to do it at the beginning and get it out and make sure we do this right. But, you know, I've had great students that were working with me in Oklahoma, again in Florida, and then now in Pittsburgh. I think really we're just you're just fortunate when we work with such talented people that they really can move you and make you perform at your best. Of course, you have to acknowledge funding and what these days we really have to acknowledge funding. So I really want to talk about retinal degeneration, but from a different point of view than probably what most people will talk to you about. There, of course, as we all know, it's really a lot of genetics behind retinal diseases, and really the diseases we're focusing on in my lab are loss of either RPE or photoreceptors or both. That's kind of the degeneration that we typically are looking at. And if you look at, this is the RetNet or database that they were putting on initially. Steve Dieger was keeping track of it. I sort of haven't updated this in a while. But really, the ophthalmologists made wonderful genetics, and they were able to identify a whole bunch of genes. If you could look at the curve of how many genes were mapped and how many loci were identified, really, there was really just an explosion of identifying mutations in humans that led to disease. These are all ocular diseases. Really, those causing retinal degeneration, about 100 genes, if you add in the disease, AMD SNPs. But even in just AMD alone, there's actually, this data is out, this is outdated, but there are at least 32 genes that have about 52 SNPs in some of the 2016 GWAS study. And they're risk alleles, they're not actually causative for disease alleles. So really we're talking about not just disease-causing mutations, but also risk of developing a disease. And that should tell us a little bit about RD, is that there are ways that you can resist degeneration, right? You can have a genetic predisposition, but there are mechanisms that allow you to resist developing that disease. So this is kind of what we've been studying. So one of the things that I don't know why that's still animated. Hate animations. So I don't know why when we look at genetics, we think there's many different ways that you could kill a cell. Some of them involve protein trafficking, ER stress, calcium overload, many triggers that would lead to cell dying. But if you start thinking about the mechanism of the cells dying, that tends to funnel down into just a handful of pathways that are really important and involved in those pathways. And it turns out a lot of them involving some calcium oxidative stress, glucose metabolism, changes in metabolic activity, mitochondrial damage, reduction of NAD levels, reduction in glutathione, redox state. And then you start accumulating damage as protein oxidation, lipid oxidation, and as well as now what I think is probably a critical factor is DNA oxidation. Once you start accumulating these, it's pretty much, touch the screen. It's pretty much going to happen. So the guy walks out and I touch the screen. So this is where we're thinking that we probably need to have some intervention before you start getting to that point, right? Once it starts going down to this very late stages, that probably Josh would probably tell us different. We just need deuterated DHA. But really, I do think this is really important that we need some interventions that we can prevent that. And then we can actually have probably we can delay or reduce degeneration that's independent of the particular mutation that we have, right? So this is kind of the thought we were trying to look from. And so why would we think metabolism is important for retina? Really, and I think, we need to think of the photoreceptor as a very specialized cell. It is a biosynthetic machine, right? About 80% of the total protein and the lipids that are in a retina are actually in that cell type, right? Really incredibly abundant accumulation of those materials requires incredibly active biosynthesis, right? Of course, the RPE has the other effect, right? They have to digest that material and remove it and take it away, so you have a balance between anabolism and catabolism in these two cell types, right? So they actually work together to maintain that environment. This is they make 10% of the lipid every day, each cell, right? This is a really incredible process. These guys are really, really incredibly active. They need a lot of nutrients and substrates to make that happen. There's really a high demand for that. In addition to that, they have what's known as a dark current. So in the dark, they're really generating a current between sodium potassium ATPases and the open channels in the outer segment. This uses up a tremendous amount of ATP to drive that. So normally on a daily basis, they need lots of energy, a lot of redox equivalents, they need a lot of substrates, and they're really just there's like a racehorse, right? It's like driving a Ferrari versus a Toyota, right? So these are Ferraris. They break down a lot, right? Unfortunately, these guys don't break down a lot, right? They can do this. There are mechanisms side that drive that metabolism that keeps them functioning at that level all the time. So it's important to understand what those mechanisms are. And of course, there are a number of studies that have shown that you can recharge or reestablish metabolic activity in these cells, and that's actually beneficial and protective. So if you increase glycolysis, the rod-derived viability factor, or through reprogramming using sirtuins, there are really some evidence that you can increase that. You can change protein regulation by activation of mTOR pathway. It really can modulate how healthy the photoreceptor is. In addition to that, so some fantastic studies, this is just one of them from Saurabh's lab, that during early phase of degeneration, there's really a strong metabolic dysregulation, right? That's associated with this degeneration. So understanding how we regulate metabolism and how we control that is going to be key to increasing resilience of photoreceptors to degeneration. So in my lab, we've been thinking about that a bit, and we're thinking about some important questions. So one of them, how do photoreceptors balance this anabolism, catabolism, and while maintaining high levels of ATP production, right? They don't have a vascular supply. In mice and humans, there's no blood vessels in the retina. So how do they get that? How do they balance that? They got to get the nutrients in or they got to make it, right? So this is what we would understand that mechanism for that. And then how is it disrupted early degeneration? And then the really important question for me is how do neurons remain alive and functioning for years, maybe even decades, despite having a mutation that's ultimately going to kill that cell, right? That's the question that we've really been focusing on for a long time. Because if we can identify those mechanisms, then we can use that to extend retinal survival even longer. So we call these endogenous protective responses. Like everyone, we use model systems to kind of study that. A couple of them we've just shown here is a P23H. It's a rhodopsin mutation. It's a knock-in generated by Chris Palczewski's lab. And this chart is to show, we should probably do this for Zoom, is to show the rate of degeneration as over time. So this is a P21. And you can see there's a pretty sharp curve for P23H, but then it slows down and tapers out. The RD10 has a very sharp curve that just goes down to almost zero right away. So these are these two different rates of degeneration. If we're going to study these mice, we actually need to collect samples and study what happens at these very early points before we actually lose cells. We don't want to study a change in cell populations. We want to study a change in molecular signals that are associated with degeneration, not just study changes in cells. So the other model that we typically use, and some of you I've talked to today are also using this model as well. It's basically a light-induced degeneration. And in the past, we've actually found that if you put mice in very dim light, there's no stress, there's no damaging, that's called the ground state. We can rear the animals or raise them into sort of mild bright light. Not damaging, it doesn't kill any of the cells, but it induces a stress state. So we can understand what happens in a stress response. But if we take the animals from dim light and we put them in bright light, that will just kill the photoreceptors right away. However, if we induce that mild stress and then put them in bright light, they're protected, right? So we can induce resilience by that stress. That was a model that we developed when we were in Oklahoma. And the other thing about light damage is we've exposed them for 4 hours here. We've actually had the LEDs in the lid. And you turn off the lights, you put them back in the dark, and the cells don't actually start dying until 8 hours after that, right? It's not an immediate response. So we can collect time points in here. We're studying the biochemical changes that occur and associated with the stress or disease or death without actually studying changes in cell population, right? So those are important concepts.
John,
yeah.
How are you defining dying at 8 hours?
So we do apoptosis assays. And so if you're measuring tunnel, for example, or caspase activation, we don't actually see tunnel positive cells or caspase activation until around that 7 to 8 hour point.
You know that cells that are tunnel positive in the light damage model, some of them actually don't die.
We also use yeah, you're right. But at the endpoint analysis, we wait a week and then we know that their cells are actually gone, right? So there is a recovery phase. DNA damage is a major part of that. And so what you measure with tunnel is also DNA repair. I could get in a whole other lecture on that. You're measuring DNA repair with tunnel staining. But the bottom line is that we don't actually see any tunnel positive cells until that 8 hours, right? And so we're actually studying not dying cells or dead cells, but cells that are deciding to die. And this is the scale if everybody's never done light damage. You know, humans can obviously go hang out at a bright sunny beach in Florida and not go blind. So we're talking something much, much less than that. That's about 100,000 lux of light. Overcast skies, 1,000 to 30,000. We're talking something like office level lights is stressful for a rodent. If you're rearing your mice, albino mice, in a normal mouse room, I can guarantee you they're under stress, right? If it's 1,200 lux in a normal mouse room, that's a stressful state, and that's going to initiate all the responses that I'm telling you about in a minute. The rest of this lecture are occurring in those mice in a normal mouse room, if it's albino, right? So we have to lower our light levels in the mouse room so that in the cage, it's about 20 lux of light in order to do these studies. And we actually have to work with the animal facility to get the room light down to about 200 lux, right? So it is important to remember the light level, how you're rearing these animals, because that's going to affect your study. And of course, humans really don't, even when staring at the sun, we don't go blind, right? I did have a certain picture of somebody actually staring at the sun, and I took that out. So one of the things that we did was we did initial RNA, like everyone, we did RNA-seq studies to look at, we actually collaborated with a couple of people to do that, to look at what happens in RD10 versus P23H. And interestingly, we see, and I can't read my screen here because I've got this in the way, let's get that out of the way. But interesting, what we see is there's a number of things that are in common. Both models, if we look at the things that are different, you can think about finding what's a unique signature for one model versus the other. We actually took the other approach and see what they're in common, what's going on between the two. And there's really an interesting finding as we see initially, we see a lot of things that are going up to say metabolism, protein transport, but seeing the gene expression like translation. We see upregulation genes are going to induce cell death or protect from cell death. It really is kind of a mixed bag of things. But we look at things that are downregulated. We see downregulation of light stimulus. Most of the phototransduction cascade is downregulated in these genes in both models. We also see we see a number of decreasing signals for metabolism, as well as decreasing in protein synthesis, right? So it is a mixed bag of things, but so it kind of gets confusing, but you look at it, somehow I'm not switching. So we actually switched over to another study where we look at RNA-seq data, and this is actually single-cell RNA-seq from a light damage study. And we took time points that are, these P60 means they're controlled, they're not damaged at all. Immediately after light damage is 0 hour, 4 hours after light damage, 10 hours, and 24 hours after light damage. And we sorted out the cells, and the gray cells here are rods, so the pink cells are cones, and the orange and the red here are Mueller cells, either resting Mueller cells or activated Mueller cells. So we can really identify what the cells are. So we bioinformatically pull out the rods, and we just say what's different between those different time points and rods. We actually see very similar pattern to what we see in P23H samples, right? So we're looking at general responses to injury and stress, which is what we're trying to categorize by these degenerative stress subtypes. Things involved upregulation of genes immediately involved in metabolism, genes involved in downregulation, genes involved in phototransduction, and just really shifting in how they're changing gene expression. But they overlap quite well with what we see in the inherited disease models. But it's really kind of interesting, because most of the common regulated genes are actually associated with resilience. Seems like it's an adaptive response to lead to protection or to slow down the degeneration, not promote degeneration. Remember, we're not studying dead cells, we're studying cells that are deciding whether to die or not. And a lot of the signatures in the gene seem to be involved in metabolism and also downregulation of phototransduction genes. Both of those two things are associated with increased resilience or protecting from cells dying, which we thought was kind of interesting. So kind of the biggest hits that we're seeing in those pathways, I'm showing them both here, just kind of give you a roadmap of where we're going with the rest of the lecture. But we see actually a decrease in the AMPK-mTOR pathway. Talk to you about that in a moment. That's going to regulate protein synthesis. That's also going to regulate metabolism. And we're also seeing an increase in this factor, leukemia inhibitory factor, or JAK-STAT pathway, particularly focusing on the STAT3 activation. We see this as another major target that we've identified in that screen. And been working on both of those pathways in the past, so I was actually quite pleased to do it. I didn't cherry pick these, and I'll show you in a minute, but these are what is the largest signature. So let's look at the genes involved in the AMPK-mTOR signaling pathway. There's about 70 different genes that you could pull out in the databases that are associated with that. And in both RD10 and P23H, most of those genes are actually downregulated. If they're changing at all, there's a big, strong signature in downregulation in both models. And if you overlay that on top of the KEGG pathway, it pretty much saturates both mTOR and AMPK pathway. Both upstream activators, here's AMPK in the middle, here's mTOR back over here. Downregulation of factors are going to lead to activation of those pathways, such as adiponectin receptor or LKB1, or looking at insulin receptor, PI3 kinase, for example, and regulation, increased expression of these TST2 and TST1. So what we're seeing is actually these pathways look like they're signatures that the cells are responding to that stress, and it looks like it's sort of tuning down these metabolic responses. So that made this pathway very interesting to us. And of course, AMPK is an important kinase. It's called adenosine monophosphate dependent kinase. And it is it's a sensor for energy in a cell. And it recognizes AMP. And when it's bound to AMP, it leads to a net activation of AMPK. And that will phosphorylate a number of substrates to increase glycolysis. It will decrease fatty acid synthesis. It will increase fatty acid oxidation. It will also importantly induce mitochondrial biogenesis and autophagy. The general role of this kinase when it's active is to decrease energy utilization through protein synthesis or fatty acid synthesis, and will increase protein or nutrient supply, so increasing glycolysis and increase in autophagy. Those are what's known in literature. It's a very well-studied enzyme. If you do a search with AMPK, you're going to find 20,000 publications easily, right? So there's a lot known about that. Fortunately, there's not many studies in the retina, so that gave us room to do some work. So we found a drug that we could use to activate AMPK. It's a very common drug given to type 2 diabetics called metformin. And this is kind of a published, so I won't go over it too long, but we found a dose-dependent protection. We gave a little bit of metformin. We could protect from light damage. Here's without metformin. Here's a little bit of metformin. As we increase the dose of metformin, we could eventually protect the photoreceptors from that light damage, right? And we thought that might be through activation of AMPK. So we checked that, and we gave daily injections of metformin. And you can see we actually started getting could accumulate phosphorylation of AMPK over time. We also, the substrate of AMPK is PGC1 alpha, and we could see stabilization of AMPK. It usually gets dephosphorylated as well as deacetylated, and that leads to stabilization. And over time, with metformin treatment, we also got activation of PGC1 alpha. That's key because PGC1 alpha is important for mitochondrial biogenesis, which is, you can measure the mitochondrial DNA content. And with metformin, we saw basically daily injection, we saw an increase in mitochondrial DNA content in the retina. One of the surprising things that we found, though, is when we actually looked at mitochondrial gene expression, we didn't really see a major increase with just metformin alone. It actually took the stress with the metformin to initiate robust transcription in the mitochondria. So it's not just getting more mitochondrial DNA there. You needed the stress on top of that to initiate that response, right? So this is somehow that stress is signaling resilience through mitochondrial transcription. And you can measure that from nuclear transcription of TFAM as well as the mitochondrial transcripts, just showing one here is COX-2, part of the ATP synthase. So really, we're looking at stress. We can stimulate the body replication. We can prime the cells to respond well, but there has to be the stress on top of that to initiate that transcript in the mitochondria. So we know metformin is important. We can do protection. We did studies. I published these already, so I don't want to go too long. But these are RD10 mice without metformin. This is the changes in outer nuclear layer thickness with metformin. We can dramatically delay the degeneration of rods, and we can completely preserve the function and structure of cones in that model, right? So metformin was highly protective. It took days of treatment, right? It took its constant daily treatment, but it was able to delay degeneration of rods and protect cones. We also disabled that RD10. We showed it in light damage. But so it really is not just one model. It's multiple models we can protect. But we knocked out AMPK specifically in the retina, and we lost the effect. So it is something that's intrinsic to the retina, right? It's not a systemic effect of changing glucose or metabolism systemically. It's something that's happening in the retina by itself. Also can protect RPE if those using sodium iodate models. We were able, so this is a sodium iodate treatment, 30 milligrams per kilogram, basically lose the RPE. And this is looking at without metformin. These are metformin and sodium iodate. So we're able to really protect the photoreceptors and the RP from these acute insults of light damage or sodium iodate, and can protect the photoreceptors from inherited diseases with that. This is just simply by giving a drug which activates AMPK and stimulates metabolism. Of course, this is my first movement into any kind of patient data, and I can tell you right now, electronic medical records are terrible data sources. They are absolutely horrible to work with, but you can get signals for this. This is actually looking at odds ratios. This is a multivariate logistic regression. And what it simply means is this odd ratio of 1 means there's no change in risk, increase or decrease. But if you have less than 1 or lower than 1, it means that you're actually decreasing your odds of developing disease. And if you're looking at diabetics, we're protected from getting AMD, from getting a diagnosis of AMD, let's put it that way. And if they were on metformin, they were also protected from getting, of course, most type 2 diabetics get metformin. So we had to do a regression analysis where we could separate the two, independent analysis. And there is a signal for metformin by itself. This study has now been repeated by 7 different groups now, so I feel pretty strong about it, even though records are horrible. They had different patients. Some are even larger than our study. We had about 8,000 patients. Other groups have like 20 or 30,000 patients, including the Million Veterans Program. So this really is, I think, a signal that if you're taking these kind of medications and changing metabolism, it is going to slow or change the rate of degeneration that you're going to see. Could skip all that. Interestingly, though, if we knock out AMPK just in the retina, we had the 2 catalytic subunits, the alpha-1 and alpha-2. We had to knock them both out. And you can see here we knocked them out in the retina using Chextan-Cree. We don't really cause a lot of degeneration. Look and see here, if this is out in months of age, we don't really see much change in thickness until about 8 months of age. But almost immediately, like when the retina is fully functional in development, we start seeing a change in function, right? So they're already losing their ability to respond to light, both A waves and B waves. And I can tell you more about that in a minute, what we think is going on there. Most of the change in thickness that you see in these later time points is actually in the inner segment. It's not loss of the cells as much as loss of the inner segment. And that's important because what we saw in EM and looking at mitochondria is these are the nice healthy mitochondria of a rod, very elongated tubular-like structures. And then the double knockouts, these are two different animals that we see, they're popped like little berries, right? The mitochondria are not forming these highly efficient structures. They're fragmented and it looks like they're undergoing maybe some stress responses there. And if you look in the cones, they're really swollen and they do much, much lower electron density, right? So it really doesn't look like there's an effect on mitochondria. And if these data down here, the graphs are summarized over here, we actually see a reduction in mitochondrial gene expression. We see decreased expression in BT1 alpha, decreased expression in TFAM, and more importantly, we see a reduced mitochondrial copy number, DNA. So without AMPK, photoreceptors don't understand their metabolic load, and they're not able to stimulate, that's our interpretation, they're not able to stimulate mitochondrial biogenesis and repair as measured by lower DNA as well as decreased BT1 alpha expression, right? So that's how we interpret that. We did what GenHi we injected C13 and we did metabolic flux assays to try to see where the glucose, is it coming in at normal levels or is it lower levels? And the bottom line is everything is reduced. So glucose uptake is lower, glucose flux into glycolytic intermediates and the TCA cycle intermediates are all reduced, right? Which is consistent with the lower metabolitic rate that we think they're having. And somehow that's affecting synaptic transmission. One of the ways may be through lower glutamate levels because you're actually reducing glutamate as well. That's, of course, a major transmitter for photoreceptors. So it looks like AMPK and maybe PGC1 alpha are both required for this high metabolic activity of the photoreceptors. It allows the photoreceptor to sense its metabolic load and respond to that robustly to initiating metabolism, right? So essentially what we're going to stop this story right quick, wrap it up, but AMPK and mTOR function as a unit. I didn't talk about mTOR, but it's actually the other side of that. AMPK actually is a repressor of mTOR activation. And so this AMPK pathway builds nutrients that mTOR needs for function to generate protein synthesis. mTOR is actually inhibited by AMPK activation, so it would decrease protein synthesis. But mTOR, when AMPK activity is low, mTOR activity is usually going to be higher because you're going to now build proteins, you're going to initiate signaling, and that will actually use up nutrients. So somehow these two pathways work together. And when we overactivate mTOR, we also get a similar effect on protection. Did that with Katerina Lobanova a couple of years ago. So really these two function as a unit, and more study is needed to understand how those things are working together. I kind of call them the yin and yang of metabolism here for that reason. So I'll move over to this part of the story. We're going to talk about the stress response involving STAT3 activation and leukemia inhibitory factor. And this is, I think, is a pathway is definitely happening in every degeneration model we've ever looked at. Actually, you just stick a needle in the eye, you can stimulate this pathway. This is a very general stress response. This is in every patient. We can see it in human data sets as well as in rodent data sets. This is not something that is just in the mouse models we studied. So a number of years ago, we were screening around looking for things that were stemmed from my postdoc days. Actually, I started working on LIF. I made a transgenic mouse to express LIF from the lens. And we always made transgenic mice at the time and FEV mice. So I was surprised. I'd never seen a photoreceptor my first couple of years of my postdoc because they didn't have any photoreceptors. They're RD1 mice, so they all lost photoreceptors. I made a mouse express LIF, and for the first time, I'd seen photoreceptors, right? And so this was in a mouse histology other than normal mice, right? But this was an example. This was an extreme event where a factor could be protecting photoreceptors from RD1, right? That mouse happened to be 12 months old. So photoreceptors typically die within 2 weeks in RD1, but that was a 12-month-old. They weren't functioning. didn't develop properly, but they were there. So I started thinking about survival of photoreceptors. So I moved away from transgenic mice because obviously that's not the way to go anymore. But we started thinking about LIF and its ability to protect. As we wanted to see, is it ever expressed on the retina? And so a normal mouse is not expressed. There's absolutely no expression of LIF in a mouse retina that we've ever measured that's normal and healthy. But if you look at this is a VPP, it's an autosomal dominant, it's a rhodopsin mutation model, it's transgenic of VPP. And as they start to lose photoreceptors, there's a massive increase in LIF expression. And as they start dying off, that LIF expression goes down. But it's not just LIF. There's LIF, there's oncostatin M, there's FGF2, there's BDNF that can also follow that same pathway. But LIF was certainly among the cytokines that we measured. Christian Grimm has also shown that in the RD10 model, it's the same thing happens. So the cells start dying off, you get this massive upregulation of LIF that will actually go down over time. And those of you who think, oh, you're just studying CNTF doesn't respond that way in mice. And I'll talk a lot about CNTF if you want, but we've actually had clinical trials with CNTF, and I think, and Gus, you're part of those for dog models, but CNTF the only cell it can really stimulate in the retina are Mueller cells. The receptor isn't expressed on the other cells in the retina. So however, LIF and GP130 are expressed. So LIF receptor and GP130, the receptors for LIF, are expressed in all cells, right? So they can all respond to that cytokine. So here are the receptors, LIF receptor and GP130, and here are the two cytokines, oncostatin M and LIF, mapped onto it. These are, I think, important for me because they're actually a gene duplication project. They're on the same chromosome in tandem, both in mice and humans. They're very close together. And they both combine and activate the same receptor complex. So we're kind of focusing on both of those. And so the receptors all lead to activation of STAT3. Once it's phosphorylated, it actually goes in the nucleus and drives transcription. So that's an active transcription factor. We can inject in LIF, and very quickly we can get phosphorylation of STAT3 in all cells on the retina. So not just activation in Mueller cells, but everything. And we've done a lot of studies to show that it really is a very potent protective factor. Christian Grimm has actually looked at knockout mice for LIF, and you can knock out LIF and it has no effect on the retina unless there's another ongoing stress. So this is actually the same VPP model. This is the degeneration of VPP. If you do the degeneration in the background of LIF knockout, they degenerate much faster. So no degeneration, just knock out LIF unless there's an ongoing stress involved. We did a similar experiment, but somewhat different. We took mice and the same mouse, and we injected in LIF. Actually, these are two injections at 14 days and at 21, and we looked at 28 days. And this is injecting a recombinant protein, and we got protection and delay in degeneration. But we also made an antagonist, which would bind one of the receptors, but not the other. So it would antagonize endogenous LIF. And that antagonist actually made the photoreceptors die faster, right? So by knockout or by antagonism, if you block these receptor signaling, photoreceptors are lost resilience and they're more sensitive to degeneration and they will die faster. So we took a couple of other models. We knocked out GP130, we knocked out STAT3, both in the whole retina, but in this case, just rods. And if you knock out those signals in intrinsic signaling rods, if we knock the receptor or STAT3 out in rods, they die faster. This is the VPP model. Here's the curve. This is the knockout, so there's no loss with the knockout. But this is the rate of degeneration in VPP. I'm sorry, this is the rate of degeneration in VPP. This lower curve is the rate of degeneration in VPP without STAT3 activation. And it's the same result with GP130 knockout. So loss of the signaling system in rods decreases the resilience of the photoreceptors, and they'll die faster from that inherited mutation. And if you look at the weeks, I mean, this is basically changing the death it's doubling the rate of cell death. Right? So we think this is a major resilience factor and in stress-induced pathway that we think is playing a role in rates of degeneration. So we went back to our single cell work. And actually, just to show you briefly, we have two different populations of Mueller cells. We have resting Mueller cells. We have activated Mueller cells. So those are the two cells we want to contrast. They're activated in this case by light. It also is true they're activated by, we looked at P23H study data as well, and they're activated by NMDA. They're activated by needle injection, right? Stress induces those activated Mueller cell responses, and they're very similar. And if we look at where we see LIF expression, it's only expressed in these activated Mueller cells, right? Pretty uniquely. And oncostatin M, which is on the same chromosome, right next to it, is only expressed in microglia. This is really a beautiful expression pattern. I swear to you, I didn't take Photoshop and erase all the red dots elsewhere, but this is really a unique expression pattern that I found very satisfying. So we really wanted to understand what is inducing that LIF, what's regulating LIF expression. So we took out those Mueller cells, all of the Mueller cells, and we was done a pseudotime analysis, right? So these are single cells, and it's how they change over time in our experiment, right? So this is, again, resting. This is 0 hours, 4 hours, 10 hours, 24 hours after light damage. And you do pseudotime, and it plots out the cells based on similarity. So these are the resting cells over here, and then based upon changes in gene expression, the gene expression shift looks like these cells are more closely related to these cells. And these cells are the most distant related from these cells based on pseudotime analysis. However, if we look at the time points, it's inverted from that. These are the resting cells in the orange here, are also, those are 0 hour and 4 hours, but some of the orange are over here, some of the orange are over here, the transition period. The cells go immediately from resting to its furthest point in gene expression. Very rapid response. Those cells are primed to do that. Within a matter of hours, they can go from 0 to 100, right? And if you think about the car analogy, right? Very fast. They go from one state to the other very fast. And then over time, they start walking back to this state, right? They change their gene expression, which is closer to the ground state. But really, there are 3 independent states. We call it the resting state, this secretory state, I'll get to a moment, and then this fibrotic state. So there's really a dynamics to photoreceptor. And actually, it turns out these can cycle through that. It's not a permanent shift. It's all based on regulation of gene expression. So what are in those different states? So LIF, one of the genes we've been looking at, is in this very highly secretory state, but so are there very prominent genes involved in inflammation, CCL2, other genes involved in inflammation expressed by Mueller cells at that state. GFAP, however, is in this 3rd state, this fibrotic gliotic state, right? So secretory cells are very transient. They come up, LIF comes up, and then it comes down, and then you turn on that fibrotic state, right? So this is kind of the dynamics of Mueller cells. What's driving that? How do Mueller cells respond to that stress state to drive that expression, right? So we were looking at, and some of you have been involved in this. I think, Brent, you might be related to some of this over here. So some people have been thinking about these protein oxidation, lipid peroxidation reactions. These are actually ligands for innate immune system. These are ligands for toll-like receptors. They're actually called dangerous associated molecular patterns, or DAMPs, right? So part of the immune system reacts to bacterial pathogens, but it also reacts to damage signals. And we're thinking that this might be part of that mechanism. That we see all of these DAMPs that are upregulated with stress, either with light stress for hydroxynonenal, or CEP, either with light stress or with these in mutations that are so, and AMD, this is an AMD patient. So essentially, these ligands for toll-like receptors are made by photoreceptors and other cells in the retina and can activate toll-like receptors. So we wanted to investigate that a little bit. So we wanted to know if toll-like receptor necessary for induction for that stress response. Think about it, if that stress response is activating toll receptors, if we knock out TLR2, we might lose that stress response, right? And I wouldn't be standing here telling you about this if it didn't happen. So we actually published a few years, I don't go too much detail here. But in the absence of toll-like receptors, we lost our LIF induction. It was dramatically reduced. And other markers for that stress response is hemoxygenase 1. And we saw a decrease in the amount of phosphorylated STAT3, right? So toll-like receptors seem to be an important part of that LIF induction. So the logic is, if that's important for that resilience, then they should be more sensitive to stress-induced damage. And that's actually what we saw. So if we look at light damage, and this is undamaged mice, untreated, and if we just do light damage on wild type or the littermates that deposit their toll-like receptor controls, this is the light damage we get with them, but if we take the knockouts, the toll-like receptor knockouts, we get more damage, right? We don't have as much LIF expression, and now we've lost some of the resilience for the resistance to degeneration. So that means it's necessary, but is it sufficient? So we took bacterial cell wall components called Pan3 cis, we injected that into the mouse eye, and we did that protect them from light damage. So is it sufficient for inducing that mechanism? And the answer is yes. Our data says it likely is. And so this is a Pan3 cis injected to light damage. We had nice ERG responses. We don't get them if we inject in PBS. So we have something above just sticking a needle in the eye, and you can measure that both by ERG as well as by measuring outer nuclear layer thickness. So activating toll receptors is not only necessary but sufficient to induce that protective cascade. Of course, who wants to go and inject bacterial cell wall components in their eye weekly? That's probably not a good idea. So we're just thinking about this as how do we study that pathway? So we went back to our single cell data. Which cells make the toll-like receptor? This particular is toll-like receptor 2, which we think is the receptor for these DAMPs, these lipid peroxidation DAMPs. That's only expressed over there in those microglia cells. Pretty unique. You can, however, see it. You guys can't see it so much there, but you can see it in some of these activated Mueller cells. And it's those very early Mueller cells that are secretory. They can respond to TLR as well, so you can get somewhat of an application. But resting Mueller cells don't express it. It's just expressed in these microglia cells. So now we're talking about those stress neurons talking to microglia directly, right? Can microglia respond to light damage? The answer is yes. Here's what microglia look like if you stay in them with the IBA1 antibody, which you can see the cell body, and you can see these ramified processes. That's a normal process for microglia. They're just kind of sniffing around looking for damage. You hit them with light, hit the retina with light damage, and yes, they're going to find damage. They're going to become amoeboid. They're going to migrate to that site of injury. You see they've lost the processes, and now these cells are actually able to move and migrate. If you knock out toll-like receptor 2, they're much less efficient at losing that less efficient at becoming in the amoeboid state, and they're actually not able to migrate nearly as effectively to the sites of injury. So they lost the signal for that response. So we think microglia are part of that signaling to induce lift through some factors. So microglia get activated toll-like receptors, and now they're going to signal to Mueller cells. How do they signal to Mueller cells? So we went back to our single cell data set. We started looking for ligand receptor interactions. We had beautiful results with LIF and oncostatin, and we thought, well, maybe we'll do it again. We did. So if we look at IL-1 beta and TNF alpha, they're expressed exclusively in microglia by the RNA level. And if we look at the receptors, IL-1 beta receptor is not expressed in Mueller cells, but in mice, but it is in humans. But if you look at TNF alpha receptor, it's only expressed in Mueller cells, astrocytes, the blood vessels, the RPE, microglia as well. So it's really limited to what cells can respond to microglia, right? Microglia come in, they get activated by toll-like receptors, they release TNF alpha and IL-1 beta. The cells with the receptors can respond. Mueller cells are very likely to respond to TNF alpha. In fact, they are. We can inject in TNF alpha, we can induce LIF expression, right? So we're thinking we've working our way down that pathway for that resilience. And we know they're responsive to TNF alpha because they can induce activation of NF-kappa B, which is a major signaling component for TNF alpha receptor. So we've got a nice progression. Really is a beautiful ligand receptor study in terms of their interactions. It fits well. And so what we're thinking now is that Mueller cells are able to go from this resting state into this highly secretory state through TNF alpha secretion and activation of NF-kappa B. And in fact, we know now that LIF, based on single nuclear ATAC-seq, as well as some epigenetic studies, that LIF and CCL2 are highly dependent on FOS activation and NF-kappa B, right, at the transcript level. So they actually bind the promoters for those genes, and those elements are necessary for driving their expression. We take two enhancers for LIF, and it's definitely NF-kappa B, or actually TNF alpha responsive, and they're very potent enhancers. So they're at least as strong as SV40 early enhancer. However, that's not true for GFAP. GFAP is actually STAT3 dependent. If you knock out STAT3, you don't get GFAP expression. And so what we think is happening is that LIF is being induced by TNF alpha for Mueller cells, and Mueller cells are inducing LIF because of TNF alpha response. LIF is secreted, it then signals back onto Mueller cells and activates STAT3, and that's what's driving this gliotic fibrotic state. There's a loop there. And this is a completely reversible process. That could decay down, the TNF alpha goes away, the LIF expression will go away, the STAT3 phosphorylation will decrease, GFAP expression will decrease, and they'll go back into resting state. So this is not a fixed expression. This is a dynamic expression. In fact, Michael Dyer has just recently put a paper by archives talking about the dynamic responses of mural cells, and they seem to be an incredibly dynamic cell. They are a lot of prime promoters, just like LIF. So the promoters are primed for expression. All it takes is a very small signal, and they'll get rapid induction. So there really is some really good fit with other people are finding as well. So in summary, I think we're getting close. So this is kind of our model for that damage response. I think we have these damaged cells. They're actually releasing ligands for toll-like receptors. Toll-like receptor is one of many toll-like receptors. I think we can actually think about toll-like TLR4, and possibly if you have release of mitochondrial DNA, you can also think about an RNA. You can think about TLR9 as well. I think there are other toll-like receptors that are participating that we just haven't really dug into yet, but those will activate all are expressed in microglia, by the way. We think that's one of the ways that microglia respond to injury. Any of your disease models, this is taking place, right? How many of you have studied microglia migration in your retinal? Yeah, okay. Think this is probably how this is getting activated. We think TNF alpha is a key player. We're testing that now through knocking out TNF alpha as well as the receptor, particularly Mueller cells. But we also are looking at the transcriptional changes initiated to the ChIP-seq or cut and run assays for NF-kappa B in specifically in Mueller cells. And what we're thinking now is that that is necessary for production of LIF. LIF, we think, can come back and antagonize some of these NF-kappa B promoters. We'll discover that pretty soon. That's our current hypothesis. And that's part of the mechanism for what shuts it all off again in Mueller cells and how they can go back to that resting state. So LIF is terminating that more of that secretory phase. In photoreceptors, though, LIF can go and activate STAT3, and that goes on to promoters that can increase the expression of those resiliency factors that we've been studying. We're looking at STAT3 dependent binding in the promoters for genes and rods specifically, as well as how they might interact with NRL and CRX. A little spoiler note, we found that STAT3 seems to be able to downregulate NRL expression. And so that might be one of the ways that all the phototransduction genes can be tamped down, not turned off, but can be reduced when you activate STAT3. So that's kind of where we are, I think, at that pathway. So we found these two major pathways. We don't think they're all of them. We think there's more pathways we can look at and study. I think there's a lot more going on than just this, but we think these two pathways in particular are really regulating retinal resistance to degeneration. Both of them are changing in every model of disease that we've ever looked at. I don't want to go out and say it happens in everything yet, but I think this is probably a background. The things I'm talking about are things you probably are seeing in your own data when you're looking at retinal diseases. And I think this is something that we all ought to talk about and try to categorize what's real specific to one disease versus what's a general stress response initiating that resistance and resilience. With that, I'd like to conclude with this slide. Scan that QR code. We are actually organizing the next RD meeting if you're interested in retinal degeneration. We're going to have it in this beautiful city. You might have heard of it, Prague. Assuming that we all are still working and living and we can still go, but let's look for the positive. I think we could go to this meeting and have a great time, and we can really focus our interest on retinal diseases. So thank you very much.
Questions?
That was really great. If you look at a mouse model of, rapid versus slow degeneration, does LIF kind of follow those dynamics? And do you think there's a difference in acute injury versus chronic injury in these pathways?
I see it as more, I think it's somewhat differently. So I think it's responding to the injury to the cell, right? So, and actually, if you look at by in situ where LIF is expressed, it's pretty focal. It's not every Mueller cell, but it's near where the cells are dying. Right? So if you have a very slow degeneration, you just have fewer cells that are dying, so it'll be fewer Mueller cells that will get activated, right? If you have more cells dying faster, so more rapid, then you'll have a more robust response. But it seems to be pretty focal, and it's because of the microglia, it's highly localized to where actually the cells are injured. And of course, I think that correlates with where the microglia migrate to, right? So they actually can get activated, they migrate to that sites of injury. Then they're going to start releasing the TNF alpha and IL-1 beta, right? So it becomes very focal at that point. So the more broadly you have it, I didn't mention it, but the same pathway, the same toll-like receptor, TNF alpha, and activation of STAT3, those are also the same signals that zebrafish use for regeneration, right? So this isn't something that evolved to protect the retina. This is actually a response of immune cells to a lot of things. And in zebrafish, that's programmed for regeneration, whereas in mice, that's programmed for this induced protective state, right? So it's somewhat different in terms of thinking about it, but evolution just changed that same signaling pathway for two different purposes. So fish tend to replace photoreceptors, and humans tend to protect them, we think. So, and in fish, when you have very slow degeneration, you don't induce regeneration, right? And I think it's the same answer. You don't have enough of the signal taking place to initiate that regenerative response, right? If it's so small and so low and so focal, it's not large enough a signal to get that to initiate a regenerative response. That's a long answer, but
So that's what I was getting at, because the slow one doesn't regenerate. So can you just overload that with LIF and then help that process?
We tried LIF actually with David Hyde, and it turns out the LIF receptor is somewhat different enough in fish that we can't use human or mouse LIF. We can use CNTF in fish, and that will initiate that same regenerative response. So,
does these pathways differ between rods and cones and photoreceptors in the center versus the periphery, or are they pretty much everywhere?
And does it differ with age? So a lot of diseases are associated with aging and declining in that resilience with age, right? These are exactly the right questions that I think we're trying to get to. The gene expression regulated in cones is different than in rods, although I would say we don't have enough data on cones yet to know for certain, right? So it's still a work in progress, but I suspect the gene expression is definitely different in RPE than rods, and it's definitely different in Mueller cells than in rods. And I suspect that we're going to have some differences in cones as well.
And mouse cones are kind of funny anyway, right? Yeah. Go ahead.
Yeah, Josh.
Great talk, John. So do you think LIF could be used therapeutically, or would it be
so over the next hour?
So you'd be inducing a stress response chronically and downregulating visual transduction genes. And so could LIF be used, or would you want to use something downstream of LIF in photoreceptors potentially?
Yeah, that's what we started doing some of the gene expression studies. We wanted to look what was downstream, but what we see is that it's multiple pathways that are being stimulated, right? So STAT3 is activating not just one thing, it's activating like 5 or 6 different pathways to initiate that protection. And so far, we haven't found the one thing downstream that's really going to be it. It may be the conglomerate of all of those, right? So we're looking at things like constitutively active STAT3, which was a disaster, right? That's too much, right? However, we have injected large boluses of LIF, and the maximum we can suppress the phototransduction gene is about 2-fold, right? So it's not turning them completely off, but it is downregulating them about 2-fold, right? So I don't know so CNTF in RP patients was ended because patients were reporting no real benefit and actually some loss in sensitivity and vision, right? So that was thought not to be a great result, right? So we don't know if that's going to be different or not. So, but the problem with CNTF is it's only stimulating Mueller cells and making Mueller cells doing something different. So I don't really know what that is yet. But also we're making, I think the levels matter, right? How much you would reduce that. And so the Neurotech device had certain levels, but patients got what they got, right? So we're making a gene therapy. We've made a gene therapy and tested in rodents that you can actually regulate with a secondary drug, right? And so it was basically a stabilized LIF that we could do the gene therapy. And then there's no LIF expression, no detectable LIF expression until we get the second drug. And now we get LIF protection, right? Then we could get LIF media protection from the RD10 in the RD10 mouse model, right? Sodium iodide, light damage, the whole thing. So we can regulate and titer that. And in those mice, we can have it low enough to get protection, but not high enough to cause loss of detectable function in rodents. I think that approach might work, but if not, we'd like others. I'd like maybe work with Gus or William to kind of test these in dogs at some point. We just need to move forward with that.
Hi. Thank you for the talk. Yeah, I had a question about, I guess, other things that might induce stress And I don't know if it's relevant for eyes, but if exercise has some kind of systemic effect, like, is that?
So you might have read the papers. There's actually, what's his name in Emory, Jeff Boatwright, has been using stress-induced protection. And actually some other people doing that as well. And they're linking that to BDNF induction. He's looked for LIF. He said he didn't find it, right? So they're thinking this might be more of a systemic induction of BDNF, which is complicated by itself because only the inner retina has BDNF receptors to BDNF, right? So that's kind of a complicated for that. But there are people who are actually studying things about exercise. But exercise will also do something else, and that will change metabolism, right? And there's a very real possibility that you're actually changing the AMPK activation through these decrease of metabolites that you're getting from this exercise. That has not been addressed, right? So that's a very, come to Pittsburgh, we can study it.
Go ahead. And then you have a question in the chat also.
John, you mentioned that in slower diseases, when you have foci of degeneration, then you have LIF expression in Mueller cells.
Just in those foci.
In the foci.
Yep.
So if you look at the TNF alpha, is it also expressed in the same cells?
TNF alpha is expressed in the microglia, and it's usually expressed in the microglia to home in on that dying cell. So that's usually where you're getting the TNF alpha expression.
So they will be then in the outer.
No, they migrate into the outer. They migrate to the outer nucleus. They're in the outer plexiform layer. But when you have a damaged photoreceptor or even a damaged RPE, they'll go all the way to the RPE. They're really mobile and they can move all the way down to the RPE. So, in fact, you're looking at some foci that form some microglia moving to the RPE layer. Yeah.
We have
a chat question. Cyrus?
Yes. Thank you for your talk. I was just wondering if similar to like LIF, if you see any pattern with other IL-6 cytokines like oncostatin M in terms of them being secreted from microglia, and then but secreted from Mueller glia and then acting on photoreceptors and RPE.
So funny thing about that is I talked myself out of doing the LIF knockout mouse and Christian Grimm did it instead because there's like so many different cytokines in the same family. You knock out one, you could have redundancy. In his study, there wasn't a lot of redundancy. So LIF knockout was pretty extreme. However, we see oncostatin M and cardiotropin-like cytokine, they're both the same family, are expressed in microglia and induced by that stress. Cardiotropin-like cytokine could also be expressed in Mueller cells. But based on Christian's work, I mean, it looks like there's not a lot of redundancy between those different family members. And IL-6 can't replace LIF as well. So IL-6 is also part of that family. And it doesn't seem to be able to complement the loss of LIF. But there's still room for studying that question, though, I think.
Okay, thank you.
Mm-hmm. Okay.
There's no more questions. Thank you again.
Thank you. Bye.